When we talk about germ layer evolution and then also evolution of the coelom, all of those things are going to be happening over here at the base of the animal clade, at the base of Uniconta. And so our most ancient member of Uniconta are going to be the Periphera, which you remember are the sea sponges. They're more of a colony sort of a structure. They don't have true tissues. Once true tissues evolved down here, Ctenophora is the first member of the animals that actually does have true tissues. And the way those are going to look is that, remember that when we have something like a blastula, the blastula is going to be this group of cells that are going to be found in a sphere, kind of like this. You also might see drawings where it looks like this, where we've got a layer of cells that surround a cavity, and the cavity that is to the inside is the blastocele, but the entire group of cells together is the blastula. And this is this hollow ball of cells that is going to be right after you'd find the morula. And again, the reason why they have to be hollow is that way the cells that are on the periphery can get rid of waste and also have room to respire and take in more nutrition. The next stage that will happen is that the blastula is going to go through gastrulation to form a gastrula. And remember that the gastrula has got an opening down here at the bottom, and then it has two layers of cells that are going to make up the gastrula, and those two layers of cells are going to be formed when the bottom of the blastula moves upwards, kind of pushes in like this, which is going to make this hole. And now these two layers are what used to be surrounding the blastocele inside of the blastula. So to indicate these two layers of cells, I'm just going to draw some little division marks here just to show where those cell membranes would be. And when we get to the gastrula, we've got this opening right here, which is the blastopore, and then we have these two layers of tissues. We got the endoderm and we have the ectoderm. And if you're talking about something like Ctenophora, the sea combs, and then also Cnidaria, the sea jellies, this is really their entire body. They've just got an endoderm, they have an ectoderm. If we're talking about a sea comb, the Ctenophora, this is really their entire body plan. If we're talking about something like Cnidaria, and they're a medusa form, for example, then they're going to look like this. And the two tissue layers we see over here in the gastrula, those are the two tissue layers that they have inside of their bodies. And so a Ctenophora and Cnidaria are basically just a swimming gastrula. When we get over here to Acelamate, which is this guy right here, by the time we get to Acelamate, we have the evolution of a mesoderm, and now the organism is triploblastic. And so those cells that are going to be in between the endoderm and the ectoderm, which are the mesoderm, right here, when they first evolve, we're just going to have an area that is completely solid with tissue. All of this is going to be full of cells, and so this tissue layer, the mesoderm, has no empty spaces in it. It's just packed full with tissue. And again, this is going to be what we find over here with Acelamate. And so when it very first evolves, this is what we call Acelamate. And so Acelamate means that all of this area is completely full of mesoderm. And because it's completely full of mesoderm, we really can't have very much complexity because there's no room for anything new to evolve. When we come over here to a pseudocilamate, that means that the mesoderm has kind of retracted away from the GI tract, from the endoderm, and so the tissue is still there and it's still packed densely, but there is an empty area right here surrounding the GI tract. And so this is this is the mesoderm, just like we saw over here in this picture. But this cavity that is just formed, this is called the pseudocoelom. And so it is an empty space where we can start to have new new organs evolve within this empty area. But once they do evolve, let's imagine that, some kind of new structure shows up right here. There's going to be two problems. The first is it might push against the GI tract over here, causing blockages or problems inside of the organism. Also, it only could attach over here to the mesoderm. it doesn't have anywhere to attach over here on this side, which means it could be, kind of knocked loose or rattled loose, and that'd be a problem. The other thing is that now our GI tract is just kind of bouncing around inside the organism. There isn't anything supporting it. And so although we've gained this pseudocoelom, this empty space, so that we can potentially have the evolution of new organs, we now have potentially taken a step backwards in that the GI tract isn't supported. So the best compromise is to have a truly coelomate animal. And what this means is that we have these empty areas within the mesoderm where new organs can evolve. And that's what this is. These two things that look like the letter C. That would be the coelom. And our mesoderm tissue is right in here. And so the benefit of this is that now, because we have this coelom, we have space for innovation. We could potentially start to evolve things, like a heart. Maybe this is going to be the heart. Maybe this is eventually going to evolve to become the liver. We can have these new organ structures evolve. When they evolve, there's areas where they can become attached right here. But at the same time, our GI tract, which is the endoderm right here, it's supported by these like bungee cord looking things of tissue that are made up of the mesoderm. And so the mesoderm would be right here. So with this kind of configuration of a coelomate animal, we have the best of all possibilities. We've got area for innovation. You can get new organs. When the organs evolve, they have places to attach to the mesoderm, and our GI tract is still supported by mesoderm in the middle, just like we started with over here in an acoelomate organism. But we have room to grow, and that the coelom is actually there. So this is kind of the weird thing, is that when we say the evolution of the coelom or a coelomate animal, it makes it sound like you're gaining something, and you are, but the thing that you're gaining is just empty space. And that's what this is, the coelom. So since we began over here on our tree in Aceoella with something that was triploblastic, but at the same time acoelomate, all three of these groups, both the deuterostomes, the lophotromes, and the ecdysosomes, are all independently, on their own, going to go through these evolutionary steps that are going to go from acoelomate to pseudocoelomate to coelomate. So the transition to those three body plans is going to happen more than once, and they're going to be analogous in those three clades.